Jah would never give power to a bald head. Run, come, crucify the dread. Time alone, oh, time will tell. You think you're in heaven, but you're living in hell. Think you're in heaven, but you're living in hell. Think you're in heaven, but you're living in hell. Time alone, oh, time will tell. You think you're in heaven, but you're living in hell. Back them up. Not the brothers, but the ones who set them up. Time alone, oh, time will tell. You think you're in heaven, but you're living in hell. You think you're in heaven, but you're living in hell. Think you're in heaven, but you're living in hell. Time alone, oh, time will tell. Time alone, oh, time will tell. Time alone, oh, time will tell. You think you're in heaven, but you're living in hell. For children, weep no more. Oh, my sycamore tree saw the freedom tree, saw I settled the score. Children, weep no more. Weep no more. Children, weep no more. Já jamais permitirá que as mãos do terror venham sufocar o amor. Somente o tempo só dirá se irei luz ou permanecerei pó. Se encontrarei Deus ou permanecerei só. Se ainda hei de abraçar minha avó. Somente o tempo só. Time alone, oh, time will tell. Somente o tempo só. Time alone, oh, time will tell. You think you're in heaven, but you're living in hell. Don't worry about a thing. Every little thing is gonna be alright. So don't worry about a thing. Every little thing is gonna be alright. Rise up this morning, smile to the rising sun. Three little birds pitch by my doorstep, singing sweet songs of melodies pure and true. Say, the 'cause every little thing is gonna be alright. Rise up this morning, smile to the rising sun. 3 little birds pitch by my doorstep, singing sweet songs of melodies pure and true. Say, this is my message to you. Você que eleve-se alto ao céu, com seus pés no chão. Eleve-se alto ao céu, que o reggae é o dono do salão. Eleve-se alto ao céu, não diga não. Eleve-se alto ao céu, em afirmação. O que você faz então? Você faz assim, como nunca fez, enfim. Você sobe assim, desce assim, canta pra mim. Você vem assim, vai assim, assim. Escanca assim, escancaradamente. Eleve-se alto ao céu, não diga não. Eleve-se alto ao céu, assim diz papaizão. Eleve-se alto ao céu, com seus pés no chão. Você que eleve-se alto ao céu, que o reggae é o dono do salão. Menina, o reggae vai lhe pegar pelas cadeiras, vai lhe pegar pela cintura. Menina, olha o reggae aí. Você que eleve-se alto ao céu, com seus pés no chão. Eleve-se alto ao céu, que o reggae é o dono do salão. Eleve-se alto ao céu, não diga não. Eleve-se alto ao céu, em afirmação. O que você faz então? Você faz assim, como nunca fez, enfim. Sobe assim, desce assim, assim, canta pra mim, menina. Vem assim, vai assim, assim. Escanca assim, escancaradamente. Eleve-se alto ao céu, não diga não. Eleve-se alto ao céu, assim diz papaizão. Eleve-se alto ao céu, com seus pés no chão. Eleve-se alto ao céu. reggae lhe pegou pela cintura, lhe pegou pelas cadeiras. Menina, 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 olha o reggae aí. Ha!
Semantic memory, what are the different separations here? What's working memory, short-term memory, long-term memory? What are the interesting categories of memory?
Yeah, and so memory researchers, we love to cut things up and
Hmm.
say, is memory one thing or is it two things? There's two things, there's three things. And so one of the things that there's value in that, and especially experimental value in terms of being able to dissect things. In the real world, it's all connected. Speak to your question, working memory is a term that was coined by Alan Baddeley. It's basically thought to be this ability to keep information online in your mind right in front of you at a given time and to be able to control the flow of that information, to choose what information is relevant, to be able to manipulate it and so forth. And one of the things that Alan did that was quite brilliant was he said, there's this ability to kind of passively store information, see things in your mind's eye or hear your internal monologue. But we have that ability to keep information in mind. But then we also have this separate, what he called a central executive, which is identified a lot with the prefrontal cortex. It's this ability to control the flow of information that's being kept active based on what it is you're doing. Now, a lot of my early work was basically saying that this working memory, which some memory researchers would call short term memory, is not at all independent from long term memory. That is, that a lot of executive function requires learning and you have to have like synaptic change for that to happen. And but there's also transient forms of memory. So one of the things I've been getting into lately is the idea that we form internal models of events. The obvious one that I always use is birthday parties, right? So you go to a child's birthday party. Once the cake comes out and you just see a candle, you can predict the whole frame, set of events that happens later. And up till that point where the child blows out the candle, you have an internal model in your head of what's going on. And so if you follow people's eyes, it's not actually on what's happening, it's going where the action's about to happen, which is just fascinating, right? So you have this internal model, and that's a kind of a working memory product. It's something that you're keeping online that's allowing you to interpret this world around you. Now, to build that model, though, you need to pull out stuff from your general knowledge of the world, which is what we call semantic memory. And then you'd want to be able to pull out memories for specific events that happened in the past, which we call episodic memory. So, in a way, they're all connected, even though it's different. The things that we're focusing on and the way we organize information in the present, which is working memory, will play a big role in determining how we remember that information later, which people typically call long-term memory.
So if you have something like a birthday party and you've been to many before, you're gonna load that from disk into working memory, this model, and then you're mostly operating on the model. And if it's a new task, you don't have a model, so you're more in the data collection.
Yeah, so one of the fascinating things that we've been studying, and we're not at all the first to do this. Jeff Sachs was a big pioneer in this, and I've been working with many other people. Ken Norman, Leila Devachi at or at Columbia has done some interesting stuff with this. Is this idea that we form these internal models at particular points of high prediction error, or points of, I believe, also points of uncertainty, points of surprise, or motivationally significant periods. And those points are when it's maximally optimal to encode an episodic memory. So I used to think, oh, well, we're just encoding episodic memories constantly, boom, 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 boom. But think about how much redundancy there is in all that, right? It's just a lot of information that you don't need. But if you capture an episodic memory at the point of maximum uncertainty for the singular experience, right? It's only going to happen once. But if you capture it at the point of maximum uncertainty or maximum surprise, you have the most useful point in your experience that you've grabbed. And what we see is that the hippocampus and these other networks that are involved in generating these internal models of events, they show a heightened period of connectivity or correlated activity during those breaks between different events, which we call event boundaries. These are the points where you're like surprised or you cross from one room to another and so forth. And that communication is associated with a bump of activity in the hippocampus and better memory. And so if people have a very good internal model throughout that event, you don't need to do much memory processing. You're in a predictive mode, right? And so then at these event boundaries, you encode and then you retrieve and you're like, okay, wait a minute, what's going on here? Rangannath's now talking about orcas. What's going on? And maybe you have to go back and remember reading my book to pull out the episodic memory to make sense of whatever it is I'm babbling about, right? And so there's this beautiful dynamics that you can see in the brain of these different networks that are coming together and then de-affiliating at different points in time that are allowing you to go into these modes. And so to speak to your original question, to some extent, when we're talking about semantic memory and episodic memory and working memory, you can think about it as these processes that are unfolding as these networks kind of come together and pull apart.
Why can't we roam this open country? Why can't we be what we want to be? We just want to be free. Yes, we want to be free. Be free. Be free. 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 Come on, baby! Sass me out. Check my life if I am in doubt. Hey, hey, Mr. Cop. What you saying down there? Hey, hey, Mr. Cop. My baby, no. Buffalo soldier, dreadlock rasta. There was a buffalo soldier in the heart of America, stolen from Africa, brought to I'm in it. When I analyze the stench, to me it makes a lot of sense. How the dreadlock Rasta was the buffalo soldier, and he was taken from Africa, brought to America, fighting on arrival, fighting for survival. Said he was a buffalo soldier, dreadlock Rasta, buffalo soldier, in the heart of America. If you know your history, then you would know where you're coming from. Then you wouldn't have to ask me, who the heck do I think I am? I'm just a buffalo soldier in the heart of America. Stolen from Africa, brought to America. Said he was fighting on arrival, said he was fighting for survival. Buffalo soldier, when the war for America. Ha! Yo, 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 yo. Buffalo soldier, trotting through the land. Said you wanna run, then you wanna hang. Trotting through the land. Buffalo soldier, win the war for America. Buffalo soldier, dreadlock Rasta. Said he was fighting on arrival, fighting, fighting, fighting for survival. Driven from the mainland to the heart of the Caribbean. Come on, baby. Yo, 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 hey, yo, 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 woo, yo, 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 ha, yo, 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 woo, yo, 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 hey, yo, 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 yo. Trotting through San Juan in the arms of America, trotting through Jamaica, a buffalo soldier, fighting on arrival, fighting for survival. Buffalo soldier, dreadlock Rasta. Yo, 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 ha, yo, 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 ha, yo, 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 hey, yo, 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 yo.
How about getting your shipping rates calculated in 30 seconds? How about quick quotes? How about a simple and efficient user interface?
In this informative video, we are going to learn about the RNA, or ribonucleic acid. RNA plays a crucial role in many cellular processes. At its core, ribonucleic acid is a molecule essential for the transmission of genetic information and the synthesis of proteins within living organisms. While RNA and DNA share some structural similarities, several key differences make RNA uniquely suited to its roles in the cell. At the most fundamental level, RNA is composed of three main components. These components are a sugar called ribose, a phosphate group, and a nitrogenous base. Together, they form the backbone and the informational code of the RNA molecule. RNA is built with ribose sugar, unlike DNA, which contains deoxyribose sugar. The presence of an extra hydroxyl group on the ribose sugar makes RNA more reactive. This extra group also makes RNA less stable than DNA. This structural difference is crucial for RNA's function and lifespan within the cell. The phosphate groups link the ribose sugars together. They form the backbone of the RNA strand. Each phosphate group connects to the 3' carbon of one ribose and the 5' carbon of the next. This connection creates a continuous repeating structure. RNA has 4 nitrogenous bases: adenine, guanine, cytosine, and uracil. Unlike DNA, which uses thymine, RNA uses uracil to pair with adenine. These bases are essential for storing genetic information. They also facilitate the base pairing interactions that underlie RNA's functions. RNA molecules are typically single-stranded. This allows them to fold into complex three-dimensional shapes. The single-stranded nature of RNA enables it to perform a wide variety of functions. It serves as a template for protein synthesis and can catalyze biochemical reactions. The single strand of RNA is composed of nucleotides, which are the building blocks of RNA. Each nucleotide consists of a nitrogenous base, a ribose sugar, and a phosphate group. The sequence of these nucleotides encodes genetic information. The specific arrangement of these nucleotides determines the RNA's function. There are several types of RNA. Each has a unique structure and function. Let's delve into some of the most critical types of RNA and their roles in cellular processes. Messenger RNA. It is synthesized from a DNA template during transcription. It carries genetic information from DNA to the ribosome. Here, it serves as a blueprint for protein synthesis. mRNA molecules are typically several thousand nucleotides long and are composed of a single strand of RNA. The length and sequence of mRNA determine the specific protein that will be produced. For instance, the mRNA for hemoglobin, the protein that carries oxygen in our blood, is distinct from the mRNA for insulin, a hormone that regulates blood sugar levels. Transfer RNA. It is responsible for bringing amino acids to the ribosome during protein synthesis. Each transfer RNA molecule has a specific amino acid attached to it. It acts as a bridge between the mRNA and the growing polypeptide chain. Transfer RNA molecules are typically composed of a single strand of RNA that folds into a cloverleaf shape. This shape includes an anticodon region that base pairs with the corresponding codon on the mRNA. This ensures that the correct amino acid is added to the growing protein chain. Ribosomal RNA. It is a component of ribosomes, the molecular machines that synthesize proteins. rRNA molecules play an active role in the process of protein synthesis. They facilitate the alignment of mRNA and tRNA and catalyze the formation of peptide bonds. Ribosomes are made up of a large and a small subunit. Each contains specific rRNA molecules. These rRNA molecules ensure the ribosome functions correctly, making protein synthesis efficient and accurate. microRNA. It is a small RNA molecule involved in the regulation of gene expression. MicroRNA molecules bind to specific mRNA molecules and inhibit their translation into proteins. This regulatory mechanism allows cells to control the levels of various proteins. It is essential for processes such as development, differentiation, and response to environmental changes. For example, certain microRNAs are known to play roles in cancer by regulating genes involved in cell proliferation and apoptosis. The examples we have discussed highlight just a few of the many types of RNA molecules that play critical roles in cellular processes. Now, let's delve into the intricate dance of protein synthesis, where RNA plays a central role. The process begins with DNA unwinding its double helix structure, exposing the genetic code. RNA polymerase, the enzyme responsible for transcription, then synthesizes a complementary RNA strand using one of the DNA strands as a template. This newly formed mRNA carries the genetic instructions encoded in the DNA from the nucleus to the cytoplasm. Once in the cytoplasm, the mRNA binds to a ribosome, where protein synthesis occurs. Here, the genetic code embedded within the mRNA is translated into a specific sequence of amino acids, the building blocks of proteins, by the coordinated action of tRNA molecules. With each triplet of mRNA bases, known as a codon, corresponding to a specific amino acid, the ribosome assembles the amino acids into a polypeptide chain. This is how a polypeptide chain is formed and protein is synthesized. While RNA's role in protein synthesis is paramount, its influence extends far beyond this fundamental process. RNA participates in various cellular functions, including gene regulation, RNA splicing, and catalysis. Certain types of RNAs like microRNA and small interfering RNA regulate gene expression. They achieve this by binding to complementary mRNA sequences. Once bound, they can either inhibit translation or promote mRNA degradation. This process allows cells to fine-tune the expression of genes, controlling protein production and maintaining cellular homeostasis. In eukaryotic cells, pre-mRNA undergoes splicing. This process involves the removal of non-coding regions called introns. The remaining exons are then joined together. This results in the formation of a mature mRNA molecule.
Salesperson, shitty prospecting tools got you feeling like, well, lucky for you.
The splicing process is intricate and highly regulated. It is orchestrated by small nuclear ribonucleoproteins and other components of the spliciosome. Remarkably, some RNA molecules exhibit catalytic activity. They are capable of catalyzing biochemical reactions similar to enzymes. These special RNA molecules are known as ribozymes. They play essential roles in RNA processing and other cellular processes. Their discovery has revolutionized our understanding of molecular biology and holds immense potential for various applications in medicine and biotechnology. Understanding the structure and function of RNA is crucial for researchers and medical professionals. This knowledge allows them to develop new treatments and therapies for various diseases and disorders. For instance, the discovery of RNA's role in gene regulation has been transformative. It has led to the development of RNA-based therapeutics, such as small interfering RNA and antisense oligonucleotides. These are used to silence specific genes involved in diseases like cancer and genetic disorders. To illustrate the significance of RNA structure, consider the story of the COVID-19 mRNA vaccines. Scientists harnessed the power of mRNA to create a new type of vaccine. They encoded the genetic information for the virus's spike protein into mRNA. This allowed them to instruct human cells to produce this protein, thereby stimulating an immune response without causing disease. This innovative approach is built on decades of research into RNA structure and function. It has been pivotal in controlling the pandemic and saving countless lives. By delving
Feel it in the one drop, and we still find time to rap. We're making the one stop. The generation gap, so feel this drum beat as it beats within, playing a rhythm, fighting against the system. Are we? I know Jah never let us down. Pulling rights from wrong. I know Jah never let us down. I know. They need the world so hard every day. We got to keep
Check, check.
fighting.
1, 2, 3,
So hard
check.
every day. My people are dying. My people are dying. My people are dying. But read it in Revelation. You'll find your redemption. And then you give us the teaching of His Majesty. For in no one, no devil's philosophy. Huh? Give us the teaching of His Majesty. For in no one, no devil philosophy. Feel it in the one drop. One stop. Drum beat. Playing a rhythm, fighting against best and skills. I know Jah never let us down. Pulling rights from wrong. I know Jah never let us down. I know Jah never let us down. Pulling rights from wrong. I know Jah never let us down. I know Jah never let us down. Pulling rights from wrong. I know Jah never let us down. I know Jah never let us down. Pulling rights from wrong. I know Jah never let us